Hello and welcome to this BioProcess International Ask the Expert webcast. I'm your host, Leah Rosen, the online editor for BioProcess International. Before we get started, just a couple of notes. This webcast is being recorded and will be made available for replay in the multimedia section of our website. We have muted the audio lines, but we welcome you to type in your questions for our speaker in the chat window on your screen. After the presentation, we will begin the question and answer portion, and I will ask our speaker your questions from the chat window. Your questions in the chat window will only be visible to myself and our speaker. So thank you for joining us today. It is now my pleasure to introduce our speaker, Aziza Mansour from National Research Council Canada.
Yeah, thanks, Leah. Well, welcome everyone to this webinar. Before we start, I would like to just give you a quick introduction about the National Research Council. So the NRC is basically Canada's RTO. And as you can see on this map, there are several research facilities across Canada. And we work on different disciplines as varied as aerospace, energy, mining, and medical devices. So what we have in common, though, is that we serve both the government and the private sector. And we work with both national clients as well as international clients. So I'm personally based in Montreal, and I'm part of the Human Health and Therapeutics portfolio. More specifically, I'm part of the cell culture scale-up team. So our mission is to produce viral vectors, antibodies, and recombinant proteins using suspension cells in large-scale bioreactors. So in these pictures, you can see stir tank bioreactors that go all the way up to 500 liters, but we also have disposable units that can go up to 200 liters. So in general, in our project, we focus on the robustness of the process, and we look for industrially relevant strategies, so for methods that we could easily implement in biomanufacturing. So this actually brings me to the goal of this project, which is to test insulin as a booster for cell growth and also for influenza production. So why did we choose insulin? For several reasons. Partly because it's already used in cell culture and it's already approved by regulatory agencies. So this is really in line with our mission, which is to look for strategies that could be quickly implemented in the industry. Also, insulin is known to activate cellular signaling pathways that are crucial for influenza production, but I will actually come back to this a bit later. So first, let's examine the effect of insulin on an industrially relevant cell line that we've developed here at NRC. So we work with a HEK293 cell line that has been adapted to suspension and that grows well in serum-free conditions. And actually, we've used this cell line for the production of a vast array of viruses, including influenza, adeno, AAV, lentiviral vectors, and VLPs. So this cell line grows well in a number of commercial media, as well as in our in-house media. So it's a media that we have specifically developed for this cell line. So in this project, we've measured cell growth in two types of insulin-free media. So first of all, that in-house media I just mentioned. But we also wanted to evaluate the effect of adding insulin in chemically defined media called CD293. So it's chemically defined, it's protein-free, and it does not contain hydrolysates, as opposed to the other commercial media that we've been using so far. So our previous attempts actually at growing this cell line in chemically defined media were not successful in the sense that we couldn't really reach the cell density that we wanted. So in the next experiment that I'm going to show you, we've added insulin every 72 hours, and we've used two different concentrations. So here are the results that we had obtained in the in-house media, first of all. So as I mentioned before, this cell line is this media, the in-house media, was specifically formulated for this cell line. So as you can see, even here, even in the absence of insulin, the gray line, we can still reach a cell density of about 6 million cells per ml. However, the advantage is that when we use insulin, we're able to reach that cell density sooner, about 3 days sooner. So in terms of bioprocessing, this is definitely an interesting attribute because you basically accelerate your bioprocess. Next, we've also performed a similar experiment, but this time in the chemically defined media. And as you can see here, without insulin, 0 milligrams per liter, the cells didn't reach such a high density. We had about 1 million cells per ml. And the cell viability remained high, though. So it's not because the cells are dying. They're simply not growing in this media. In this case, however, when we added 10 or 20 milligrams per liter of insulin, we're able to reach a cell density of about 6 million cells per ml, which is basically similar to what we had obtained in the in-house media. So in this case, we were able to basically alleviate the limitations that we had originally observed with this chemically defined media. So for certain applications, you might want to move away from hydrolysates and et cetera. This could be an easy strategy to improve your cell growth properties in chemically defined media. So actually now I'd be interested in knowing what kind of strategy you employ in your processes. So we have a poll question, and I believe you can easily just click to pick your answers. So the question is, what strategy do you employ to get the best from your processes? And you have 4 different answers that you can pick. A, you just assume that off-the-shelf commercial media is already optimal. B, you use commercial media, but then you also add supplements, additives, and feeds to improve your process. C, you formulate your own media. Or D, you develop your own platform, so meaning your own cell line and your own media like we did with the HEK293 cell line and the in-house media. So go ahead, and I'll wait just a few seconds to see your results. So if I look at the responses that we're getting, it seems like most people either just use off-the-shelf media or add feeds and additives to their media. We have only very few people who actually develop their own platform, which makes sense because it's a big investment both in money, time, and expertise. Now I would say in terms of improving your bioprocess, adding feeds or supplements, or like in this case insulin, is probably the easiest way of improving your process, and I'm not surprised that so many people use it. All right, so now I'd like to move to the second part of this talk. And here I will show you data that we've obtained when we added insulin during influenza production runs. So before I get into the data, I'd like, so first, let's examine the rationale behind using insulin to enhance influenza yield. So here in mammalian cells, you'll see that the influenza viral cycle is actually a multi-step process. And during the replication of the virus, several signaling pathways are actually involved. And in this case, you can see that the PI3K AKT pathway is actually involved in several steps during viral replication. So we've actually verified that experimentally in our cell line. So we've used an H1N1 cell line, an H1N1 virus, influenza virus, and infected HEK293 cell line. Then we've measured the activation of this pathway by looking at the phosphorylation of AKT by flow cytometry. And as you can see here in the results, we see a very strong activation of AKT starting at 15 hours post-infection all the way up to 29 hours post-infection. So now what's interesting is that insulin is actually a strong AKT activator. So we worked under the assumption that by adding insulin, we might be able to enhance influenza production by basically potentiating or increasing the response of this pathway. So in terms of experimental conditions design, we've chosen to screen several conditions using a microbioreactor with 24 wells capacity. So when cells are infected, we've also added trypsin in order to cleave and activate influenza. And then we incubate the cell for 48 hours under controlled conditions at 35 degrees. And at the end, we collect the supernatant and determine the viral titer. Now, the experiments were performed both in the in-house media and the chemically defined media that I showed you at the beginning. We used 2 different influenza strains, an H1N1 and an H3N2 strain, and we've tested 4 different concentrations of insulin. Now, actually, anyone who's worked with influenza knows that the quantification is actually really not that straightforward. So I'd be curious now to know what kind of methods you use in your processes to quantify influenza. So here you have a few possible answers, and I think you can pick more than one. Electron microscopy, which is probably the gold standard, but it's very low throughput. HPLC or flow virometry to measure the total amount of vesicles. The hemagglutination assay with red blood cells. TCID50 assay and plaque assay, which are both measures of infectivity. The single radial immunodiffusion assay, the SRID assay, and or other immunoblotting methods such as ELISA. So I will give you, again, a minute or so to respond. In our group, we probably use all of these techniques. The infectivity assays take time, so it's not the most high throughput assay. You don't get your results until you have to wait about 2 weeks to get your results. HPLC or flow virometry, we have issues with these techniques because the results get confounded by non-viral particles, so such as exosomes and other vesicles. The hemagglutination assay lacks in sensitivity, and the SRID assay requires very specific reagents that are strain specific. And then I'll come back to the immunoblotting methods that we've been using. So now if I look at your results, it seems like, see the results. Okay, so many of you are using the TCID50 and the SRID assay. So the SRID assay, I'm not surprised because that's the only one that's actually approved by regulatory agencies to release vaccine lots, so it's not surprising. Yeah, SRID assay seems to be winning. Okay. So actually, before we move on, I just want to show you how we do the quantification in our group now. So if we look here at the picture of influenza, you see that there's two proteins expressed, as you know. So neuraminidase and hemagglutinin are both at the surface of the virus. And if we zoom on the hemagglutinin protein, you see that there's a head region and a stalk region. So most of the mutations and antigenic shifts actually happen here in the head region. So each time you have a new strain, you actually need new strain-specific reagents in order to be able to run the SRID assay that most of you are using. So you can see that it's not ideal if you need to wait for the reagents each time you have a new strain, or especially in case of a pandemic, it's actually kind of a big limitation. Luckily, though, one of our collaborators at Health Canada, Shanley, has determined that there is a specific region here called the fusion peptide that's highly conserved among strains, and the level of homology is rather high. If you look here on the bottom, you see the level of homology of this region within different strains, and actually for this analysis, it used close to 4,000 strains. So what we've done then is that we've used a peptide conjugate based on this sequence, immunized mice, and then we're able to generate monoclonal antibodies that are able to recognize multiple strains of influenza. And I'm going to show you just quickly a Western blot here where we basically have two monoclonal antibodies, two lead candidates, 11H12 and 10A9. So on this blot here, each lane corresponds to a different subtype of influenza, H1, H2, H3, etc. And as you can see, all of the different strains or subtypes are recognized by either one or both antibodies. So then what we did is that we mixed both antibodies in a cocktail that we call the pan-HA cocktail, and we've been able to detect virtually all of the strains that we've tested so far, about 40 or 50 strains that were expressed in eggs, mammalian cells, VLPs, or as recombinant proteins. Now, with these antibodies, we were able to run competitive ELISAs and Western blots, but now the technique that we're mostly using in the lab is actually the dot blot assay. So this is a higher throughput than Western blot because you have 96 well capacity. It's quite easy to implement. It's not expensive at all, and the protocol is quite easy to follow. You first have to denature your samples with urea in order to expose that epitope, so that peptide sequence that will be recognized by the pan-HA antibodies. Then the samples are loaded into the wells, and vacuum is applied onto the membrane. And the membrane is then incubated with the pan-HA antibodies, followed by a secondary antibody. So let me just show you some results, an example of the results that we usually obtain. So on the top, you see an example of what the membrane looks like. And in the first two columns here, we have the calibrating antigen. So this is a standard that we had previously quantified. And you can do that either by electron microscopy or SRID if the reagents are available, or any other technique. So this calibrating antigen was loaded in concentrations between 160 nanograms per ml up to 20 micrograms per ml. And we've generated the calibration curve here under in the figure in the bottom. So now if we just use the linear range of the curve, we were able to quantify the HA concentrations of 10 different samples that were loaded in 4 different dilutions to make sure we fall in the dynamic range of the calibration curve. And the results are shown here on the bottom, and they actually make sense because the 2 samples here, S4 and S5, were actually concentrated by sucrose cushion, and as you can see, the concentration is much higher. And these results were actually confirmed also using an HA assay, hemagglutination assay. So this is a method that we've used to quantify the results that I'm going to show you now. So we've infected HEK293 cell lines with an H1N1 strain, influenza strain, in the in-house media, so in serum-free conditions, in the microbioreactor. And here you have the HA concentration that's shown on the y-axis. So this was measured by dot blood, and insulin concentrations going from 5 to 100 milligrams per liter on the x-axis. And as you can see, when we used 5 and 25 milligrams per liter of insulin, we had a 1.7 to 1.9 fold increase in influenza. So almost a 2-fold increase when we added insulin. Now what's interesting is that if you look at the total cell count at the time of harvest, we didn't see any significant difference. So this increase in influenza is not just due to an increase in cell density. It's really truly mechanistically changing something in the cells so that they actually produce a higher level of influenza. So next we've performed the same experiment, but this time in the chemically defined media. And again, we saw an increase in influenza yield of about 1.7-fold in the best condition when we added 25 to 100 milligrams per liter of insulin. This time again, there was no significant change in the cell density. And finally, we ran a similar experiment in the chemically defined media again, but with a different influenza strain, so with the H3N2 strain. And in this case, again, we saw an increase in influenza yield when we used 5 and 25 milligrams per liter insulin without a significant change in cell density. So I've shown you 3 different conditions, 2 different media, 2 different strains. And so overall, if we pull all the data together, it seems like the best insulin concentration that gave us the best increase in influenza would be 25 micrograms per ml insulin. And we obtained 1.5 to 1.9-fold increase when we used this condition. So we're already at the end. And just to summarize the data, during cell growth, we confirmed that adding insulin to the cell cultures actually improved the cell growth properties of our cell line, and 10 milligrams per liter insulin was enough. It both accelerated the growth in the in-house media, which was already optimal, but it also allowed us to use the chemically defined media, which we were not able to do before, actually. In terms of influenza production, when we use our own quantification reagents, we've determined that adding 25 milligrams per liter of insulin in our process actually led to an increase in influenza yield by almost 2-fold, depending on the strain and condition. And it's in line with the kind of processes that we look for, because this is something that could be easily implemented in a biomanufacturing process, for instance. Now our next steps are really to first confirm the results that we've seen in the microbioreactor with influenza in a larger scale. And we're also interested in exploring the effect of insulin on other cell lines and viral vectors. And just before I finish, I'd like to quickly thank my colleagues at NRC, especially Sonia Tremblay, who was involved in the technical work. And thank you also to our collaborators at NovoNordisk for supplying the insulin and for their scientific help. So that was what I had to present to you today. And I think now I will see if you have any questions. And answer your question. So,
Okay,
Leah,
great.
yeah?
Thanks, Aziza. Yeah, we have a couple of questions come in. The first one, does the higher HA relate to more virus particles?
Okay, so let me remember what we did. So I didn't do any. I did look at the number of viral particles by HPLC, and we did see an increase in the number of viral particles. However, the results are not, how can I put it? It'd have to be confirmed by electron microscopy because we tend to sometimes see an increase in particle counts by HPLC simply due to production of exosomes from the cells. So yes, I did see an increase in number of particles, but I could not conclude for sure that they were all influenza particles. So I'd need to redo this and or check the samples by electron microscopy.
Okay. And are there any results or ideas for the effect on the B strains?
Oh, that's a very good question. I have not tested the effect of insulin on B strains yet, but it's actually something that I'd like to do. My guess is that we should see a similar effect because I think the way insulin works is by activating that AKT pathway, which basically increases cell survival. So I think what it does is that it extends the window during which the virus is able to bud out of the cell. So with B strains, I'm guessing the effect would be similar to what we've seen with H1 and H3. So I have not tested it experimentally, but my guess would be that it should work as well.
Okay. And so was the insulin quantity for the best result different or strain dependent?
Yes, it seems like it because with the H3N2 strain, it seems like we needed slightly lower concentrations compared to the H1N1 strain. So it could be strain specific actually, yeah. Yeah, I'm looking at the data quickly. It also seems to depend also on the media. In the in-house media, we needed lower concentration compared to the chemically defined media. So I would suggest to try 2 or 3 concentrations for sure before starting, including that in your biomanufacturing, for instance.
Okay. And given the cost of insulin, is the addition of insulin a viable option for biomanufacturing of influenza vaccines?
I mean, I guess a cost evaluation would have to be made for each, for your system. But I would say that from a bioprocess point of view, if adding insulin increases your yield by 2-fold, that should also mean that you need to run less runs, 2 times less runs. So that's definitely a good way of reducing the cost. Also, if your product is more concentrated, it might make the next step a bit easier in terms of purification and concentration. You have to counting the amount of work and hours spent on making several campaigns. So yeah, a cost evaluation has to be made, but if you get at least a 2-fold increase in influenza by adding insulin, it's probably cost effective, yes.
Okay. Well, I think that is all the time we have for questions today.
Okay.
So thank you very much, Aziza.
Thank you.
And thanks to our audience for joining us. Again, the recorded version of this webcast will be available for on-demand viewing on our website, and as a registered attendee, you'll receive a follow-up email providing you with a direct link. We look forward to having you join us for our future BioProcess International Ask the Expert webcast. We have a full lineup covering many aspects of bioprocessing scheduled for the duration of the year. Look for those announcements in your inbox. Thanks again. Have a good day. Bye.
Thank you. Bye.